Good afternoon, or good morning, or good evening, wherever you're joining us from. I'm David Hanna, and I'd like to welcome you to our World Water Day 2021 week of events at the University of Birmingham. Since we think every day should be World Water Day, we've gone from World Water Day being yesterday, the 22nd of March, to making a whole week of events. As you'll be aware, World Water Day celebrates under the UN's observance water and raises issues around freshwater related matters. And for 2021, World Water Day is all about what water means to people, its true value, and how we can better protect this vital resource. So World Water Day is running under the theme of Water to Me. And as part of our week of World Water Day events, we're hoping that you'll be able to share your views on what water means to you through our Mentimeter poll. You can click on or take a QR photograph of the code on the screen now. And we've also put the link to the poll in the chat for today's event. It'll take about 10 minutes to complete. And we very much encourage you to play an active role in crowdsourcing information to help us think about how we might transition from Water to Me, the theme for World Water Day this year, to thinking about issues of water for all and how we address Sustainable Development Goal number 6. So back to today's event. And it's a real great pleasure to welcome our speaker for Tuesday on World Water Week, Laurent Pfister. Laurent is a hydrologist based at the Luxembourg Institute of Science and Technology, where he leads the Environmental Sensing and Modelling Research Unit. He's also an affiliated professor in environmental science at the University of Luxembourg, and very importantly, an honorary professor here at the University of Birmingham. His research interests are wide. They fit under the theme of holistic understanding of hydrological systems in the context of global change. And one hallmark of Laurent's exciting work is his innovative use of novel technologies to better understand the water environment. So he's used techniques such as thermal infrared imagery to map saturated zone dynamics and catchments, two terrestrial diatoms as hydrological tracers, and excitingly, some very high impact recent papers using freshwater mussels to reconstruct past signatures of oxygen isotopes in stream water. So we're really fortunate to have Laurent joining us today as one of the world's leading catchment hydrologists and to hear him speak on the very, very exciting topic of The Rivers Are Alive, and an intriguing title. So Laurent, I will hand over to you and welcome, and we're interested to hear your talk. Thanks.
Thank you, David. And thank you for the invitation also. So I'm going to share my screen. There we go. Yeah, so it's really an honor and a pleasure for me to give this presentation. And I always find it a bit funny that it's, of course, it's we have the World Water Day, we have the World Water Week, and for me it's always a special week also because it's my birthday. I'm born on the 24th of March, so that's couldn't be a better fit in terms of dates for a hydrologist, I would say. So today I'm first spending a few minutes on giving an introduction on well, what actually motivates us in our work in our research here in Luxembourg. And so what we are very much interested in now for almost 20 years is the way how catchments function. So really focusing at the three major hydrological catchment functions. So the way how they collect the water, how they store the water, including all the mixing that might take place. And then, of course, also at some point, this water is released. And so we basically, all our work is somehow connected to these catchment functions. And so we do, of course, have specific knowledge gaps that we are focusing on. One of them is really getting a better understanding of the mechanistics that sort of link the storage of the water and the way how it is released to the streams. And a very important aspect there is to better understand how eventually geology is sort of controlling these fundamental functions of water collection, mixing, storage, and release. And also what we have been focusing on for a very long time is also understanding how these catchments are responding, or how they are going to respond to global change. So it can be changes in land use, can be, of course, climate change. So basically, hydrological functions in the context of non-stationarity. And so here I have listed 4 research questions that we will focus on today. So what is, at least in our setting, in our study area, the relationship between catchment bedrock geology and let's call it streamflow metrics. I will say a few words about that later, what that exactly means for us. Then the second question, how does bedrock geology affect stream isotope response to precipitation and the long-term damping of isotopic signatures? And of course, subsequently on the catchment transit times. How robust are our findings in the light of non-stationarity? So that being the third question. And finally, and here I already reveal part of, so to say, the secret. So like if we want to understand how the catchments are going to respond to non-stationarity, we have a problem in that we don't have time series that exceed, say, 40, 50 years at best. And if we talk about climate change, we, of course, need way longer time series. And David already mentioned it a bit. So we have in recent years tried to find out in freshwater mussels ways to extract isotope information and thereby expanding our time series. So a few words about the physiographic context. You see here a geological map of the sedimentary Paris Basin, and the small red rectangle here in the upper right part of the map, that is basically where Luxembourg is located. So the southern part of the country is part of the sedimentary Paris Basin, so where you have different layers of bedrock types. Some are permeable, others are less permeable, and really extending over hundreds of meters. And then in the northern part of the country, we still have this Paleozoic bedrock. It's very old, very, I would say, well, it depends actually. It's not everywhere impermeable. We did the mistake for quite a long time saying that it was impermeable, but actually it's a bit more complicated than that. But so a very different substrate there. Here you see a cut through the Paris Basin, and so I don't know if you see my mouse, but so we are here on the eastern border of the Paris Basin, and so we have this contact zone between the Paleozoic bedrock and the schists and the sedimentary layers of the Paris Basin. Now, taking a closer look to where we are, so you see Luxembourg here on the map in between Belgium, Germany, France, and we have there the Alzette River Basin, where we have been monitoring now for a little bit more than 20 years, precipitation, discharge, and many, many other variables. And here we focus, especially for this storage-related work, we focus on 16 nested catchments that have different areas, so ranging from half a square kilometer to more than 400 square kilometers. Altitudes are not that different, so roughly between 400, 500 meters above sea level. But what is really making it so special is that we have a large mix of different bedrock types. So this is very much a simplification, saying that we just have schist, sandstone, and basalts. So we will see that in a minute. And of course, very important, the isotope measurements, so relating to hydrogen and oxygen in precipitation and stream water. By the way, a little bit more than 8 years now, we're more like coming close to 10 years. Just given our location, we have a very strong influence in terms of meteorological conditions by the North Atlantic. So we don't have a strong seasonality, I guess a bit what we also see in the UK. So very much influenced by the westerly atmospheric fluxes. So a quite even distribution of precipitation over the year, but a very strong gradient, and that is topographically driven. So a lot of, or let's say, way more rainfall coming down in the western part of the country, up to 1,000 millimeters, and going down to 700, just 30 kilometers further east. But when we take a look at potential evapotranspiration, we see, of course, a very different picture. Almost zero in winter, and the highest levels in summer, of course, all driven by temperature. And that then eventually has a very strong influence on the runoff. So we usually observe the highest runoff in the winter month, and low flows end of summer. Now, taking a closer look at the way how our catchments function, you see really here how these catchments operate as filters, but that these filters operate in very different ways. 3 examples here for winter responses and summer responses on sandstone, marls, and schist-dominated catchments. Just very short, when you take a look at the green line, in winter you don't see very high peaks. You don't see them either in summer. And most importantly, you have a very stable base flow, which relates to the fact that this is sandstone, and the sandstone operates a bit like a huge sponge that takes up all the water and then gradually releases that water over the weeks and months. And it's very different story when you look at the red line that corresponds to a catchment dominated by marls. Very flashy response in winter. These catchments have a very small storage capacity, meaning that they run dry as soon as we go into, say, April, May, June. These catchments run very quickly dry. And then during the summer, you only have these very local and very tiny responses to precipitation. So, and I would say the schists are somewhere in between. We don't have the time today to go into the way how these catchments functions, but they are also really fascinating. Now, the way how physiographic characteristics control runoff coefficients, we have been looking into that a long while ago, about 20 years ago. And we could see that essentially the runoff coefficients that had been observed versus the ones that had been estimated were very much controlled. We could see that already then by the percentage of impermeable substrate. Now that is also a very, I mean, pure geologist or hydrogeologist, they don't like it when we speak in those terms, because there is no such thing as a completely permeable, impermeable bedrock. But we just say that here for the sake of simplification. And so that work was done about 20 years ago, and that was what actually sort of perceived us to continue operating our catchments in different physiographic settings and really make them go and keep them running regardless of the efforts that it would take. Now that meant that at some point we were able for a wide range of catchments of different sizes, of different bedrock types, that we could calculate on many years, over many years, a catchment water balance. And actually by sort of referring the water balance, daily water balance values to a reference value that would correspond to the highest water balance level that would allow us to determine the storage deficit. So meaning that when we reach the value 0, it means that the catchment is full. So there's no storage capacity available anymore. And during summer, of course, you have a gradual, how to say, increase again of that storage deficit. So what we see here, the black line actually represents the daily value of storage deficits of about not exactly 10 years. And so the amplitude between the minimum and maximum values corresponds to what we qualify as the active storage. So the variability in storage that we can observe with the data that we have. Now we've pushed this a little bit further. So if you plot like what we did here for about 8 years of data runoff, so the daily runoff values against the daily storage deficit values, this is what you get. And you have these red dots that represent a sort of envelope curve. And everything that is the right side of that curve corresponds to individual rainfall runoff events that have been observed over those 8-9 years. And as I previously said, so you have this active storage that corresponds to the values that we have observed in terms of runoff, in terms of storage deficit that calculated through the water balance. Then also what we tried was that we extrapolated this envelope curve to near zero flow conditions. And we would qualify that then as the total storage, at least the storage that is still hydrologically connected. So it means that there could still be additional water in the catchment, but it's not hydrologically connected to the stream. The difference between total and active storage corresponding to residual storage, or what you might also say is the chemical memory of the catchment. Now, as I said, we have done those calculations for all the catchments that we have. And this is what we ended up with. If we take a look at the storage dynamics, you see that we have catchments that have a small total storage that typically would correspond to catchments with rather impermeable substrate. And there it's not surprising that the ratio between active storage and total storage is close to 100%, meaning that those are catchments that get completely filled in winter, and they very often also run almost completely dry during summer. So there's no water flowing in the creek during summer. On the other end, you may have catchments that have a high total storage value, but there the ratio between active storage and total storage is small, simply because the total storage is so much bigger than the active storage, typically corresponding, for example, to a catchment located in sandstone area, for example. You may remember a few slides ago this catchment here where you had the base flow that was the same regardless of winter season, summer season. So typically that would correspond to these catchments. Then if we take a look on how bedrock geology controls streamflow metrics, and storage, we here have a nice example in that we can see that the ratio between summer discharge and winter discharge is highest in catchments where the percentage of impermeable bedrock is smallest. So again, here, by the way, the size of the circles corresponds to the size of the catchment. So it's proportional to the size of the catchment. So there, the active storage is highest. And as we go to catchments that have a higher percentage of impermeable bedrock, we also see that storage decreases and the ratio between summer and winter also decreases. So a low seasonality in sandstone-dominated catchments versus a high seasonality, of course, in the catchments that have a small storage capacity. So they fill very quickly and then they empty also very quickly in response to rainfall. Now, bedrock geology controls on the stream isotope response, isotope signal damping, so that is nothing new. I just wanted to show here that if you plot against the flow duration curve of catchments, the isotopic signatures that we have observed in samples taken at different moments of the hydrological year, you see that depending on what type of substrate you are, you have more or less, I would say, variability in the signal, especially here on the sandstone, except when there's really a very intense rainfall event and you have a little bit of surface runoff that might take place close to the stream. You have a little bit more variability, but for the rest of the year, it's a very stable signal. The catchment that has sort of 2/3 of sandstone and 1/3 of marls, it's not a surprise. You have a sort of intermediate signature, very stable signal in the low flow part of the flow duration curve, and then into the higher flows you start to see a little bit more variability. Now that brings us already to the relationship between the percentage of impermeable bedrock, so the bedrock type, and the mean transit times that we have determined with the sine wave damping method. And there you again see that the higher the bedrock permeability, the higher also the mean transit times, and then vice versa. As you go into the less permeable bedrock, you also see that the transit times tends to decrease quite significantly. Now, that is what we have observed with data that span about 10 years, sometimes 15 years, depending on the catchments. But we all know that we live in a non-stationary world, and many of the models that we employ today have been developed under the assumption of stationarity, and we know that that is not true. I already mentioned at the very beginning that in our catchments we have this very strong influence of atmospheric circulations coming from the northern Atlantic, bringing very humid air masses, temperate air masses, and so you see here on the right graph, so that is a paper that is now some 20 years old, but already then we could see how nicely actually the winter discharge is connected to the amount of rainfall that is brought in our region with these westerly and southwesterly atmospheric flows. And so every change in those atmospheric circulations might also impact, of course, the way how our catchment respond to the incoming precipitation, something that has already been very nicely shown, of course, also for the UK. And David, you have done work on that. And well, I think I skipped this because we don't have that much time. What I would like to also mention is that we observe now in the last years more and more these extreme events that you would usually find in the Mediterranean region, but not in our countries, not in our catchments. And this is typically the flash floods that we have observed on half a dozen occasions over the last, say, 4 years, which is really, really unusual. And so that brings us to this question. So to what extent, to what degree are our catchments resilient when it comes to global change, regardless of climate change or land use change, for example, that we would take into consideration? And what we did, so this is work in progress, what we did is that we tried to plot our catchments into the Bodico framework. And so without going into all the details, what is interesting to see here is that depending on the level of active storage that you have in your catchment, they would plot either below or above the Bodico curve. So meaning that, so if we simplify this, so we have on the vertical axis very clearly the geology that controls the catchment storage, and which in the end then also determines if a catchment will plot above or below the Bodico curve. So if I go back to the previous slide, so in those catchments where the active storage is highest, you won't run that easily and that quickly into any problem of, for example, drought, whereas in the catchments that plot in deep blue, those are the catchments that are very sensitive and where we could, in the near future, really observe more frequently severe drought problems. Now, all of this, and I already mentioned it at the beginning, brings us back to the fact that we have been observing the way how our catchments function, at least in the amount of detail that I have shown until now. We have been doing that for, say, 20 years, 20, 25 years at best. And in some areas, we have up to 40, 50 years of data. But, and we do have actually precipitation information, so precipitation data going back 150 years, but nothing comparable in terms of stream flow. And the same holds, of course, also for the tracer data series. And that is a major difficulty, of course, when it comes to investigating like how our catchments might have responded in the past or might respond in the future to changes in climate, for example. Now, the example that I will give now relates to the stable isotopes. And so you are most certainly all familiar with the method of measuring the isotope signal in the precipitation and then observing how the signal may or may not be damped, the degree of damping that you observe in the stream flow, and from that you can infer the travel times of the water with more and more sophisticated methods in recent years. But one important limitation is the fact that we have these short records of isotopes in the stream water. And so we did a very, very simple, a very quick literature overview about 2 or 3 years ago. And we could see that usually the length of the data series that you find in literature rarely exceeds 10 years when it comes to isotopes. And there's a few exceptions. So, for example, in Vienna, there's one series that spans now about 50, 60 years. And there already you can see that there is, of course, the annual variability that we are all familiar with, but there's also more than that. And the interesting thing is, of course, to understand where that variability comes from. For example, here in the 80s, we can clearly see that something happened over there. So that's why, in the end, we came up with a sort of crazy idea and one that if organisms, and in this case, freshwater mussels living in stream water, could possibly be a sort of a natural archive for isotope signals in water. So some interesting facts about freshwater mollusks. So we have around 1,200 bivalve species around the world, and they can filter, depending on the species, up to 50 liters per day. Some of them live up to 200 years, some only 1 or 2 years. On average, we can say they live up to 10 years. And it's among the longest lived organisms on Earth, so up to 200 years, as I just said. So a priori, they have an interesting potential for complementing the limited stream water isotope records. Now, the idea behind is that they form successive layers of calcite or aragonite. And so you see them when you take an electron microscope, you can see these tiny growth bands. And the tricky thing is to get into these growth bands and extract the information from them. And this is even a closer look. So you see the longer, I would say, bands, they correspond to the summer season, and then the finer layers, they correspond to winter season, because of course they limit their growth during the winter season. And it's also why they have a different shading, because during winter might be more interlaid with organic matter. And so this was actually the same. So we did preliminary investigations because, as I said, the difficulty is to get to this information. And so the idea was to use secondary ion mass spectrometry, so also called SIMS. And if we would manage to get that information from that kind of instruments, we would also have to compare actually the data that we get from such an instrument from the signal that we observe in the stream. And then a last question that we investigated was to see if this method, if we would be able to show that it works in our rivers, would that then also be the same, would the method be valid under different climates, different physiographic settings? So we were fortunate enough that we have, in the north of the country, we have a growth station for freshwater mussels, because many of them are endangered species. This is a European project where they try to grow them and then put them back into the environment. And about 40 kilometers away, we have our Weierbach catchment. I think Julian already talked about the Weierbach catchment some time ago. So we have the rain gauge, we have a stream gauge, so we have the stable isotope signatures for several years there. The only limitation that we had here, since these observations had been made independently from each other, so we didn't know exactly when the shell that we got from the mill had actually died. So, but still, we could still do some preliminary investigations. So this is what the beast looks like, the secondary ion mass spectrometer. And basically the idea is that you hit the sample with a primary ion beam, then you sort of generate sort of mini explosion, and you have these secondary ions that then go into a chamber where you have an electromagnetic field, and depending on the mass of your secondary ions, you will be able to detect how many of those ions you get. And so typically this is what happens. So you have here the growth bands, and then you hit with the ion beam these growth lines. And each time that you hit it, so there's a little bit of material that is liberated, and that goes then into the machine. And so these are the original images. So it's really like a burned crater into the material. And so this is those were replicas that we also had to make, just to be sure that we always observe the same signal. And so we had 79 spots analyzed. And very important also to know some sort of limitation of this method is, so the signal that you see in the shell is not identical to what you see in the stream. So because you need to apply a temperature correction factor. So depending on the temperature of the stream water that the shell is living in, you will have to apply a correction that is more or less, I would say, important. So this is what we observed in the Weyerbach catchment in terms of isotopic signal in precipitation. So those are the bars, and then the dots correspond to the signal we have in the creek. And then on the right side, those were the measurements that we had on the different growth bands of the mussels. So of course, it's difficult to extract immediately the information from a figure like that one. So that's why this is way more easy to analyze. On the left, on the box plot on the left, you have the signal in precipitation, so no surprise, there's way more variability. Then in the middle, we have the signal that was measured in the stream water, really the samples taken in the stream and analyzed with a laser spectrometer in the lab. And then on the right side, the stream water signal as inferred from the pearl mussels. So again, as I said, you must not forget that the mussel was taken from a different creek than the one that we used for the stream water signal analysis. But still, it's the same climate, it's the same geology, and we have a sort of quite similar signal in the sense that the signal that is in the shell is also damped in comparison to what we see in the precipitation. And also the average and median values are not that different from each other. So what we then did next is that we tried to find in literature examples of studies where people had worked on freshwater mussels for different purposes. None of them was really focusing on hydrological aspects, but nonetheless, those were studies where we would have the isotopic signature of oxygen in the shells and also in the rivers or the creeks that they had been sampled from. And also, so you see here about 9 different climate zones, Köppen-Geiger climate zones, so nice diversity from where these shells had been taken. And so you see here the relationship between especially the observed and estimated stream water oxygen isotope signal and latitude. So it's not surprising that you have a strong difference between the lower and the higher latitudes due to the fractionation effect, quite obviously. Also interestingly, we have the signal in precipitation that is quite nicely, I would say, matching the signal that we see in the shells. And this is the results that we get from this literature overview, from this sort of meta-analysis, even though it's not a real meta-analysis, but sort of, and without the temperature correction, because we were lucky enough to also have temperature data of the stream water for most of these studies. And so if you do the correction for the temperature of the stream water, you see how strong this relationship actually is. And this is what really motivated us then to pursue the effort. And we were quite lucky to get in touch with Bernd Schöne from the University of Mainz, who is a sclerochronologist. And he had a PhD candidate a few years ago who had been working with freshwater mussels, so Margaritifera, so that's the ones that can live up to 200 years. that had been sampled from little creeks in northern Sweden. And so he was able to bring that to a completely different level, in that he was actually able to extract almost 200 years of information like shell growth. He was also able to extract a proxy for stream water temperature, because you can imagine we didn't have stream water temperature for that for such a long period. And all of that eventually leading to the reconstruction of these stream water isotope signals. He, as you can see, he also is working on carbon-13, but here we focus on the blue lines. And that is quite fascinating. So on this graph, those are still the growth bands. So this is not really a timeline. Those are growth bands that you see here. And it's only ones that, and that is a very long procedure actually, and you need a sclerochronologist to translate the size of the growth bands into individual years. Because some of them might be very large because conditions were very favorable. During other years, conditions were maybe very rough, and so the shells wouldn't grow that much. And so there's a lot of work related to that. And especially here in the lower graph, we can see almost 200 years of stream water isotope signals. So for the 018 reconstructed in those Swedish catchments. Now the question is, what can we do with these signals? Because if we would apply the same protocols, so to say, the same methods as we do in our catchments over the past 10, 15 years, we also would need the signal in precipitation, and we don't have that. But what we were able to do was to see to what extent the signals that we see in these Swedish creeks, how well they match, or maybe not, signals in the atmospheric circulations. So we compared them to the winter and summer North Atlantic Oscillation Index. And there again, we were quite positively surprised by the rather strong, or at least encouraging, relationships that we had. So here's one example between this reconstructed stream water data 018 values and the North Atlantic Oscillation Index for the winter season. So that was quite encouraging indeed. But again, we still are here in the very early days. So we are really just scratching the surface, I think, of the potential that really lies in these organisms and in the way they could help us to really gain a better understanding of how our streams, our rivers may have responded to climate fluctuations, climate variability in the past. So all of this would of course not have been possible without the help of my colleagues Nuria Martinez Carreras, Christoph Hissler, Julian Klaus, Gwenaël Carrère, who is not anymore with us here at LIST. He went back to France, Mike Stewart, Jeff McDonald, Bernd Schöne, the sclerochronologist from the University of Mainz, Nathalie Wahl, here from LIST, so the expert on secondary iron mass spectrometry, Claire Graf, a student from Strasbourg that did this meta data analysis, and together with Jean-Nicolas Beisel, also a professor from Strasbourg. So thank you a lot for listening, and I'm happy to take the questions.